So welcome back, everyone, after lunch. So we'll have the first of two talks by Karen Alim. Karen Alim is at the Max Planck for Dynamics and Self-Organization in Göttingen. She's also moving to the University of Munich as a professor, and that's probably the reason why she couldn't make it here in physics. All right, so Karen, I don't know, we can't see your presentation yet.
Yeah, I'll change it. I just wanted to have you see my face before we switch to the presentation. So
I'll prompt you whenever there's a question, so you can respond to that.
Yeah, exactly. So I just wanted, thanks so much for the possibility and also for setting this up so I'm able to give this presentation, although I couldn't make it physically. And I would very much encourage you to ask questions whenever they come to your mind. And we'll go along. So the lecture today will be set up such that I will give spend about half the time to give you a PowerPoint presentation, and then I will switch to Blackboard and sort of provide you with the theoretical physics background a little more. And again, ask me questions whenever they come up. So now you should see my presentation, correct?
Yeah.
Perfect. Okay. So today's lecture is going to be on the physics of flow networks. Flow networks are a very crucial building block of life in the form of for example, the vascular networks of us pervading our body, the vein networks in leaves, or in a very pure form, if you look at fungal networks, you also have an organism whose only body plan consists out of a flow network. And this is just a small representation of the flow networks that we might be very familiar with. But if we, for example, zoom into the human body, there's a plethora of flow networks, whereas only the circulatory system is the one that we're very used to. But for example, the kidney has a nephron network, which is basically another flow network similar to it. There's a lymph system. There's our intestinal tract. There's lots of flow networks. It's a key building block of life because it enables you to transport resources along long distances. And so it's a hallmark of extended organisms. What is striking in these systems that in all systems, if we look at the small scale tubes that these networks are built of, is that we have the same dynamics of the same physics holding there, namely of laminar flow. So there's no turbulence, there's just laminar flow in all these tubes. So despite the organisms being very, very different here, the physics at these small scale networks is the same. And so you might be wondering if that's really true for our arteries, because our heart is pumping, so there should be a pulsatile flow. But in fact, what happens if you start from your heart and you go further down the tree of your musculature, there are muscles surrounding your big arteries, and they sort of bit by bit take out the pulsatile flow, so that in the end you really have pretty steady laminar flow in these small scales, like in the other systems. Now, what is also striking about living flow networks in particular is that they are adaptive. So they change their morphology. And so this sort of thinking about how they can be dynamic in their structure, I want to ignite one idea in your minds that's going to be sort of in the background of all these lectures that we do. And that idea is that if you zoom into a small tube in these networks, that just by the presence of flows, the flows pervade, transport information. Due to the presence of flows, the flows exert a shear force on the tube wall. The flows come with a hydrostatic pressure that is acting on the tube wall. Or the flows can transport particles, signaling molecules, with the flow. Now, we're looking at these dynamics in a setting where the tube walls are alive. And so the tube walls may now respond to this information. So now imagine that we have a tube and say there is an increase in shear force. And the tube responds to this information of an increase in shear force by dilating the tube wall. So by dilating the tube, I change the flow of this tube, right? Because the inflow might still be the same, and so the flow changes, and hence the information conveyed by the shear force changes because also the shear force changes. Now, the key is that's not only true for the one tube that I'm looking at, but if I zoom out again into a network, and I look at the dynamics of an incompressible fluid, the flows are globally coupled. And hence, by dilating a single tube, I actually change the flow throughout the network. And hence, also in a very distant tube, the shear force acting on the tube wall will have changed because the one tube dilated. And so you can see that, but just this one thought, that the flows are globally coupled, because we're looking at an incompressible fluid, and the tube walls responding to any kind of information that the flow transports by pressure, shear force, or really signaling molecules, these networks can self-organize. And how? That's very much a physics question. So because like solving these globally coupled flows is very much physicsy, and here the challenge is to find sort of the mean field description to understand how this feedback may unfold. Although there's no straightforward way of how you can actually do mean field. And so when I'm now embarking on the question of how flows and network morphology are coupled, I would just like to highlight a few terms that may come up to your mind. So things that play into this feedback of flow and flow network morphology is first of all flow control. So how are flows organized within these networks? Then what is the impact of adaptability? With adaptability, I'm referring to having a single tube that is dilating. So if that single tube dilates, how big is the impact on the other tube, on the other tubes due to this response? I'm also asking, what is the role of topology? So what is the role of the loops, the amount of loops in these networks? How does it affect this feedback? And in simpler terms, but more geometric, arises that what's the role of hierarchy? So with hierarchy, I'm here referring to having in the same network tubes that have big diameters and tubes that have small diameters. So there's a hierarchy in the diameter distribution. What impact does this have? And so this is like just setting a scope of what terms are important for this feedback. And now I would like to take you on a journey to get to know my model system with which I'm trying to answer this question, and teach you a little bit on flow control. So the challenge of identifying the physical principles of the feedback between fluid flows and flow network morphology is that this feedback is dynamic. So you want to be able to really quantify flows and network morphology at the same time. And this provides a really hard experimental challenge if you think about your arterial system, because it's somehow embedded in this tissue, and there's bones, and there's like all sorts of things that are obstructing your clear view on your vasculature. And so very much in the spirit that the physics in these small scale networks is the same fluid physics, and I'm here using a very different model system that makes this quantification very easy. And so the model system, you actually see it here in these small icons that I've shown you, and they're all from my group. So the model system I'm using here is Physarum polycephalon. Phasarum is a slime mold, and slime molds are a very ancient form of life, sort of really going back very much to the where the tree of life spread out into fungi and animals. And this particular slime mold is an acellular slime mold, which means that it goes, as the biologist would say, a syncytium, which means that I have a single giant cell, and this giant cell is built of multiple nuclei that are enclosed in a single envelope. And really, to appreciate the term giant, this cell that you see crawling around in arena with where the black dots are food sources of like bacteria droplets, this cell is about 1 centimeter in size. So you have a single cell that's 1 centimeter in size. And the way this works out is that you have multiple, like thousands, tens of thousands of nuclei engulfed in a single cell membrane, and this cell membrane also happens to have a network shape. And so here you can beautifully distinguish the reticulated network that is sort of sparse at the rear and very dense in the front, and even more dense at the front where it is growing and foraging around to find these food sources. So a little more on like how this system is set up. So you see it on the right, where you see the movie of zooming into these tube networks, and it's a purpose a little bit out of focus so that you can appreciate that the tube walls are rhythmically contracting. So what we have there is that individual tubes are built out of, so outside you have the membrane, and then inside you have a gel-like phase. And in this gel-like phase, that's very rich in actomycin. So the same building blocks as your muscles. And so what now happens is that this actomycin is rhythmically contracting. Typically, about every 100 seconds is one contraction cycle. And so in the course of these contractions, the fluid that's inside the tube, surrounded by this gel-like phase, is going to be displaced. And so because it's being displaced, it has to flow somewhere. And so you may now appreciate that there's a flow that's changing direction every so on. So every 100 seconds, this is sped up in time a lot more. So you see that the fluid flows in one direction, stops, reverses, and flows in the other direction. And so what you can also appreciate here, that really on already on this level, I can see the dynamics of the tube, and I can see the fluid flow at the same time. And now I can really embark on the question how the flows and morphology act on each other. As a first step, now, like thinking about this model system, the question that arises is like, if I have this network and all tubes are rhythmically contracting because they're all aligned with this actin cortex, the question is how are these contractions coordinated? And so to embark on this kind of questions led us to do these kind of experiments. Here, I should have mentioned like this data set where you saw the organism crawling around. You should appreciate that this took place on a timescale of 3 days. So we really sped up the dynamics of the organism a lot when I showed you this movie. Because now if we look at this data set, this is now a data set of an organism on a timescale of only 3 hours. And so on a timescale of 3 hours, you see that the network is rather static. It does not move yet. You see a little bit of like growth happening up there, but more or less the network is static. And so the way actually we prepare this is we have the organism likes to grow in a humid environment, so you grow them on an agar plate, and then you can have like big networks form, and you can trim the network to whatever size you appreciate. And so you see this a little bit, that the organism had been trimmed on these sides. So that's why you have a very specific shape. When you trim the network, basically the wounds, they heal instantly. So this fluid cytoplasm inside, it's very rich and active, and it instantly gelates once there's a mechanical stimulus. And so these tubes basically instantly heal, and then we can look at any network of our desired size, shape, whatever might be of our interest. And so you can see that this is also very nice if I want to now study the impact of topology or hierarchy, because I can choose a network that's suiting my hypothesis. And so now the way we analyze the data is in the following. So we have this big network. So you can see that in these big networks, given that the tubes are contracting every 100 seconds, and we want to measure the contractions, there's no way we can zoom in at the same time to also measure, to really measure the diameter of these tubes very well. So the trick that we use is that the amount of light transmitted through an individual tube is inversely proportional to its contractile state. And so, the key is that really the amount of transmitted light to measure this intensity. This is just a pure limitation of a camera, and you can just get a very, very good camera, and it's not like you're not limited in the dynamic range of the time changes that you can resolve. Whereas in pixels, you would be constrained if you want to measure the diameters. And so we can now go in and we can, at every pixel, we can measure the amount of transmitted light, and thereby identify the contractions of each tube. And so we started out by asking, how are the contractions organized across the network? And so now to find out how the contractions are organized, I'm going to map out to you the phase of the contractions, which tells me if an individual tube segment is sort of in the relaxed state or in the contractile state. And this is what we see. This is now showing you the phase of the contractions going from 0 to 2 pi. So it's a periodic map. And you may appreciate that you see a traveling wave traveling from the bottom left to the top right, sweeping the organism. Surprisingly, this map even matches the wavelengths that forms of the traveling wave, matches organism size. And this is true for any size of the organism. So what we can also do is cut out a piece of the network here, and it, as I said, it instantly heals. So I'm basically taking this data set, acquiring all these bright field microscopy images to do this analysis. And then I'm taking out my specimen, I cut out a piece here, and I plug it, and it instantly heals, and I plug it in back into my microscope and I measure it again, and I find that the traveling wave has reoriented along the new longest axis of the specimen, but it still statistically matches the size of the network. And you can play this game over and over again, and basically keep on cutting, and you find that there's a contraction wave forming that scales with the size of the network, that always matches the size of the network, irrespective of network size, going from an organism that's about 2 centimeters in diameter to one that's 2 millimeters. So across really orders of magnitude. And that on its own is a very interesting question to ask myself. You know, we said that these contractions are coordinated, are driven by actomyosin, that is rhythmically undergoing contractions. So somehow these contractions talk among each other across the scale of 2 centimeters to keep each other in check in a certain time-phase relationship. And just that observation is very striking. And it gives rise to very interesting theoretical work that sort of was ignited by this observation that I won't have time to, like, in depth tell you today or tomorrow, but that is published and you can read on. But I will, in the end, actually tell you the key ingredient that comes to how these coordination how these contractions are coordinated. Rather today, I would like to ask the question: why would you coordinate your contractions, and why would you generate the flows that we observe here? So when I was referring to these phase patterns, I was saying that I'm seeing here a traveling wave of contraction. So actually, traveling waves of contraction are a very renowned building block of life. We give it a specific name, and the specific name is peristalsis. So peristalsis is, for example, the way of how we transport food from our mouth to our stomach. What we have is we have one tube, and the tube contracts, and the contraction travels down, and that happens periodically. And this is the way of transporting matter from one place to another. So here in these networks, we also saw these traveling waves, but the system is, in one key aspect, fundamentally different. Namely, that the networks you saw are closed. So there is no pool like your mouth and another pool like your stomach, but the network is fully closed. And so what does it mean? It means that fluid volume has to be conserved within the network. And so the way you can generate this is, say you have one tube that is contracting, then you must have somewhere else in the network another tube that is expanding at the same time, and hence taking up the fluid volume that's displaced there. And so basically, by keeping contractions in check, you actually can conserve volume and still generate flows. And so let's just exemplify this in a very simple graph, which is like we take a single tube. So this is now a tube, and we go along the tube over time, and I'm just mapping out to you the phase of the contractions. And so the way I've now patterned the traveling wave is that basically, if you take the bottom line, you see that the phase, the same color code as before, exactly goes from 0 to 2 pi. And thereby, I ensure that if I go exactly from 0 to 2 pi, that for every tiny segment of this tube that is contracting, I have a partnering tube that is shifted by pi so that it's expanding at the same time. And so fluid volume is conserved. Now, having these contraction patterns and being aware of that we're looking at laminar flow, so at low Reynolds numbers, so the equation that are ruling the dynamics of the flows are Stokes equations. So that's sort of the simplest equation in physics that we can use. And so we can solve for the flow, which is shown here in the middle, and where red is flow to the left and blue is flow to the right. And so if you position yourself in the center of the tube now over time, you see exactly like this shuttle flow that we've observed in the data before, right? So you see flow to the left or flow to the right, flow to the left. It's changing every now and then. So that's comforting, right? That matches our expectation. We take the idea of a peristaltic wave, we put it in the simplest setting we can think about. This is a single tube, and we do find a shuttle flow. But I worked on looking in these physical mechanisms of how these flows are generated, asking myself, what are these flows good for? And now if I look at the flows in this single tube, it's a little bizarre, right? Because basically what it means, if I start here in the center and I'm a small particle, it means that the flow will take me to the left in half a period, and then it will take me just back to where I started half a period later, because it's a shutter flow. So basically this flow doesn't seem to be good for anything, because you just like, I mean, you might travel quite a distance, but you're back 50 seconds later. So it might not seem a very smart idea to put in so much energy in like constructing your actomyosin networks to generate these flows. And so this is where the key message of today's lectures comes into play, which is we're here looking at long, slender tubes. And hence the dynamics of transport can be well described by a very simplified equation called Taylor dispersion, which we are going to derive in the blackboard part of this section. And so the essence of Taylor dispersion is now, we are looking, this is the result that we're going to derive, and we're looking at the concentration of particles, but not just on the concentration, but we're looking actually on the cross-sectional average concentration, which is what I'm denoting here by the bar over the C. So I'm here showing a simplified equation for the transport of particles in these flows, averaged over the cross-section. So it's only depending on time and on the axis of the tube. The radial component has been averaged out, like actually approximated out. And what we see is that the transport of these particles has an advection term that is proportional to the flow velocity. So that's a term that doesn't do any good, right? Because it just brings me forth and back. But then I have the diffusion term, where in addition to the molecular diffusion here, I have an extra term that's proportional to the flow velocity squared, the radius squared, divided by the molecular diffusivity. So, and here you see the result, that namely if I start with a cloud of particles on the far right, you see that these particles are actually super quick in spreading out in the flow. And it's not due to the advection, because you see that sort of like the core is basically staying fixed and going back and forth, but you see that this cloud is rapidly spreading out. And it's because of this enhanced molecular diffusivity that in essence arises due to the flow profile in the tube. And by having large flow velocities, you generate a large gradient of flow velocities across the tube. And so by increasing the gradient of the flow velocities, particles may diffuse between the different streamlines along the flow, and thereby diffuse faster within the tube. As I said, the details of these equations will follow promptly, but let's just stay with these pictures in mind to think a little bit more about, now why would these contraction patterns be useful for an organism? And so I believe the first question that comes into mind is, we saw that the traveling wave always matches organism size. So why would this be particularly important to have a traveling wave match organism size? And since we have this simpler setup where we can sort of think about these dynamics, let's look at these pictures one more time, but now compare them to, this is the same as before with a single period, and now I'm adding an additional row where I'm actually going for 2 periods. So this is like going from 0 to 2 pi to 4 pi. So I have two wavelengths matched within one single tube. And so you recognize it, you see this by a steeper gradient of the phase pattern. Again, I can calculate the flow field, and already the flow field arising due to 2 wavelengths is sort of giving away what might be happening if I'm not matching the wavelengths to organism size. Because what you see here is again the shuttle flow popping up. But in the center, you actually have a region where the flow velocities are basically close to zero. And also the magnitude of the flows here in the two halves is not as high. And the key is now that I have a zone of basically zero flow velocity, it sort of acts as a border for the spread of particles. Because now if you look at the spread of particles, they're only spread out due to this enhanced molecular diffusion. So the flow velocities are really important in the spreading of the particles. But now I have a zone where there is no flow velocities. And so the only thing that can get me past the zone is really the air molecular diffusion, which is very slow. So this alone tells me that heuristically it is very useful to have a single wavelength matching organism size, because this is how you create the largest flows if you care about the transport of particles. And indeed, you can, in the simple case of a single tube, you can even solve analytically for the transport timescales, and you see how it breaks down more and more when you increase the number of wavelengths. For now, we sort of looked into the dynamics of a peristaltic wave in a single tube. But the traveling wave patterns that we've seen, they actually lived on a network. So what changes if I now actually want to put these contraction patterns on a network? Can I generalize peristalsis that I only know for single tubes to a network setting? And so the biggest constraint that I first of all have to keep in mind is, again, conservation of mass. And so the way to do it is actually to think along the following lines. I take the whole network and I discretize it in like small segments. So say, I don't know, 10 pixels in length. So I just chop it down, basically to the capacity that my code or my cluster that I have available is happy to deal with it. And now I deal with every single segment as a homogeneously contracting tube. So I just take a tube and it's rhythmically contracting. And so since this tube is rhythmically contracting, it generates a flow delta Q. And now conservation of volume means that all tubes have to be in check, so that the amount of flow generated throughout all these segments n in this network sums up to zero. And so if I actually assume that all tubes have the same diameter, the same amplitude, the same frequency, and so the only thing between these tube segments that's different is the phase of the contraction, this is actually very easy to solve because it gives rise to the sum over the exponential of the phase being to zero. And that's a very well-known solution, because that's basically adding up angles on the unit circle in a complex space. And so we know the solution to this equation, which basically means that the phases have to be partitions of the amount of tubes. So if I have, like in a simple solution, if I have n segments, I discretize my network into n segments, then my phases should be 2 pi over n, 2 times 2 pi over n, and so on unto 2 pi. And so if I just choose the phases to match this, then conservation of mass holds. And in principle, you can actually dig deeper into this mathematical problem, because you can also take partitions of the amount of segments. Like if you have 10 segments, you can also partition 5 plus 5 or 4 plus 6. And that would give rise to multiple periods. So this is a way of how I can conserve volume. But now I don't have a traveling wave. Now I just have to make sure that my contractions are actually conserving volume. But now if I want to pattern them down, I can do this in a Monte Carlo method. So now I'm taking the network that we actually observed, I discretize it into small bits, and I calculate the phases as I just shown you, and I just randomly pattern on the phases. This is by all means no traveling wave. And then I run a Monte Carlo code where I basically optimize for the local phase difference being smallest throughout the entire network. Given that I'm very much aware that these have quadratic boundary conditions, the phases, right? And so if I run Monte Carlo and I minimize the local phase difference, I actually do end up in a setting where the phases are patterned very much reminiscing what we observed in experiments. So you have a phase gradient really matching organism size, and it also self-aligns along the longest axis. Because this is what really minimizes local phase difference. This will always select for the longest axis. And now that we have these phases, we can, again, like in the example of the single tube, we can go back to the single tube concept and also calculate the flows in these networks and the transport of particles in these networks. And this is just to show you a movie of that. This is now, again, the network. I patterned out the phases, and now I'm going to run a simulation of the spread of particles where I'm also sort of, just for the visual effect, I'm highlighting which the contractile or expanded state of the tubes. And the amount of particles is going to be inoculated somewhere here. And you now see how rapidly, within just a few contraction periods, the particles, these inert particles, are spreading out across the entire network just by Taylor dispersion. So just by generating these large flows, you can drive the transport of dispersion of particles. Now, I cheated a little bit. I cheated a little bit to make math easy. And that's, I mean, that's just as a lesson. That's often good enough, right? So you may see that this cheated version was published in PNAS in 2013. Although at the time I was also aware that we cheated. And so the realm in which we cheated was that this simulation assumed that all tubes have the same radius, which we know is not true. So let me, because there is a hierarchy, there are bigger veins. And so the question is, does it matter? And as I said, I cheated, I was too lazy, I didn't do it, but then there was a beautiful, very gifted student who actually took on the challenge to check if the hierarchy of diameters actually matters. And so that was Sophie. And here, bear with me, this is the same network as before. It's just rotated by 90 degrees. You see it? This, and now we rotate, and it's the same network. Oops. So here, we're going to compare, again, the same kind of simulation, but ones where all tubes have the same radii, and I'm comparing it to the actual radii of big and small ones that we see in the actual experiment, and the total mass of these networks is kept constant. So this is how we coarse grain the radii. And so let me run the simulation on the left, and you see it like beautifully how quickly it's spreading out. Now, if we start from the same location, but we take into account that there are small and big veins, the dynamics do look different. And you see how the particles are spreading much further in the same amount of time. So this tells us that the hierarchy actually does impact the transport in the network. And as this was basically the starting point for a project for Sophie, and in the end, what we did, we investigated really in the biological specimen how does the hierarchy and the topology impact transport. And so the way to address this is that we took a network, this one, and we let it to starve for 3 hours. Because it was starving, the network self-organized, adapted its morphology. And you see it sort of in a very drastic form where tubes being pruned away in the center, which, by the way, is something that's very, it's also going on when you're vasculature forms in your brain when you're an embryo. You have a very reticulated network that you start out of the arteries that over the course of time prunes into a new structure. And so we took these two structures to ask ourselves, how well is this structure to transport particles throughout the network? And this maps out basically the effective diffusivity from any point in the network. So you see that if you start at the boundary, flows are smaller, so you don't get as far as when you start in the center. But the drastic effect comes into play when you look at how this effective dispersion increased when the network reorganized. And it reorganized such that overall the dispersion went up 40%. And it basically did so not so much using the change in hierarchy, but even more drastic change in the topology. And so basically by taking away all these country roads here, highways form, and these highways serve as highways to transport particles throughout. Due to this topological change, the fluid can only go along these big veins that left, and so the flow velocity along the big veins is much higher than previously when it was very reticulated. And that's what's increasing the Taylor dispersion. It's really by ramping up the flow velocity by pruning away the windy roads. What is an interesting effect that is not like if you were just to look at the flow velocities, you wouldn't get the full pictures. You actually have to calculate the transport dynamics, because you see that only when you also increase the flow, when you look at the transport dynamics, that not only the big tubes are important, but also being next to a big tube helps you. Because, I mean, that's basically the idea. If you live close to a motorway, you're quickly on the motorway, and so you can travel quickly. But this you only see when you also take into account the diffusion in the small roads, because you can actually really quickly get to the big roads, and this also helps you to get transported throughout the network. And so and I'm glad we solved this. So the key aspect really is to take away that we here have a flow network that is generating flows that are targeted to transport inert particles. And it's actually really reorganizing to increase the transport of particles. So transport seems to be very incremental, very instrumental for these networks. And so that's why I'm now going to derive with you the dynamics of transport in networks. So, you yell at me if I'm not writing big enough. So we're going to derive the transport of inert particles in long slender tubes. So, we have some inert particles, and we're going to describe them by a concentration C. And to describe the dynamics, they're in general described by a diffusion advection equation. That in its general form looks like dc dt plus u gradient c minus kappa, the molecular diffusivity, nabla squared c equals 0. And here we have assumed that there's no sources and no sinks, and we have an incompressible fluid. And so if you look at this equation, then it's hard, right? Because it's we have one time component, and then in space there's 3 components in general. Okay, so that's a hard task. You can't solve it. Like you would need advanced numerical schemes to do so. However, if we actually look into long slender tubes, it's much easier. And so that's the idea of Taylor dispersion, which I not only chose because it's very important for flow networks in general, but because it's also a really nice example of what I believe is good training in fluid physics, of thinking about scales and dominating terms in whatever dynamics you're looking at, which is much more important in general as an idea to look at whatever your problem you're looking at. You might very likely end up looking at a PDE that in general you cannot solve. So this is basically the same thing. So we have a PDE that in general we cannot solve, and now we're going to look at what's really important to this PDE to arrive at a setting where we actually can solve it very easily. And that's Taylor dispersion, which goes back to G.I. Taylor in 1959. No, older, sorry, 52. And so that's basically dispersion in long, slender tubes. So let's remind us of the flow dynamics in a long, slender tube. So this is my tube. My tube, first of all, is cylindrical. So if I think in polar coordinates, I get rid of the angular variable free of choice just by the symmetry of the system, and I'm left with the radial component and z along the tube. And so a tube that has a diameter a. And so now, in this tube, I have a flow profile that's called Persoid flow profile. There we go. Which means that I have largest flow velocity in the center, and there's zero flow velocity at the boundary. It's called a no-slip boundary condition, that the fluid, due to friction, has to be zero. The flow has to be zero at the very boundary. So that's my flow field U. Sorry, flow. And the flow field you might have seen derived before is 1/4 mu. Mu is the viscosity of your fluid, proportional to the pressure gradient along the tube, and then R squared minus A squared along the tube. Now, just to get training and taking cross-section averages, I'm going to simplify this equation by writing the flow velocity as a function of the cross-sectional average flow. So we're going to derive the cross-sectional average, which is U bar is 1/pi A squared, 0A, 2 pi R, and we're going to add these. And so this simplifies this as well. And then I have, so basically both terms are proportional to R. So the term, the aberration at 0 doesn't contribute. So I'm only left with 1/2 mu A squared and dpdz. And I have the fourth A to the 4 minus an eighth A to the 4, which gives rise to minus 1 over, sorry, I used the 4, so I had put the 2 in front. So it's 1 minus A squared 8 mu dp dz. And so this allows me to write now my flow field in a simpler form, where u is now u bar, so the cross-sectional average flow velocity, times 2, 1 minus r over a squared log z. And that's a much easier description. And now, if we plug in this into our original diffusion advection equation, it already simplifies a bit, right? So, because then we have the CDT, we recognize that the flow only has a direction along the tube. So this will then be u bar 2, 1 minus r over a squared dc dz minus kappa. But here, unfortunately, I still have the radial terms in it. So it looks like we're almost there, because the advection term simplifies, but actually the diffusion term is still including radial terms. And so we're not there. And so for the following, I'm going to abbreviate this as just u for saving some writing. And so now the key trick behind the idea of tailored dispersion is that I'm going to separate both the flow and the flow velocity and the concentration of particles as the cross-sectional average and the variation around it. And the same for u. U bar plus variation around it. And so the idea behind that is that I want to look at in the limit eventually of long slender tubes. I will assume that the tube radius is so small that diffusion will readily average out any changes in the radial variation, so that in the end, actually looking at the cross-section average of the concentration is a good approximation for the dynamics. And then looking only at the dynamics of the cross-section average allows me to write down an equation that only depends on the axis along the tube. And that's the key idea. So a little bit of math required, because we can actually now calculate u tilde, which is u minus u bar. So we can use that from above. This is our u minus u bar. So we deduct 1 u bar. So we end up with u 1 minus 2 r over squared. And we also have to take into account our boundary conditions, because they're going to help us simplify. Which is that the particles cannot penetrate the wall. So the flux of inert particles on the wall R equal A has to be 0. And so if I now separate this out, it means that bear in mind that the cross-sectional average of the concentration does not depend on R first place. So the gradient is by definition 0, but it sets a constraint for the variation of the concentration, namely now the exactly same, that this is 0. And that's going to help simplify it. So now I'm quickly going to, because we're going to deal with very long equations, I'm going to prematurely clean the backward a little bit. Okay, so now what I'm going to do is just plugging in this answers into this equation. And that's our equation number 1. So we have now these. Question?
Go ahead. No, go ahead.
Okay, so the C bar dt plus the C tilde dt plus U bar dc bar dz plus U tilde. Now you see why I'm using two boards, because it's actually going to extend quite a bit. Z plus U tilde dc tilde dz plus minus kappa. So the cross-section average of C only depends on Z, right? So I can save some space here. But just I have this term, but then the variation depends on Z, and so here I actually have to take those terms, and I'm actually spelling out in radial coordinates. Equals 0. And now that's the full equation. So right now I didn't make any approximation. I just plugged in these two terms. And now I'm going to take the cross-section average of this equation. And so this helps, because first of all, we know that the cross-section average of the variation by definition has to be 0. Because if I take the cross-section average of this term, then it should be equivalent to C bar, right? So I know that C tilde bar is equals 0, and the same is true for the flow field. And so this already tremendously simplifies my equation. So I keep this term. This one goes to 0 because of the C tilde. This one stays. This one goes to 0 because of the U tilde. This one goes to 0 because of the C tilde. This one actually stays because here I have variation in U and C, and they might be coupled. So I actually have to take this term, unfortunately, with me. This one stays too, because it's already C bar. This one simplifies as well, because it's only the C bar, and it's a derivative in Z, and I'm taking the cross-section average in R, and they so I can swap, I can take the Z in front, so this is 0. And this one is actually more complicated, so this we should actually work out. I'm going to work this out over here. So if we now actually calculate this term, it's 1 over pi a squared, 2 pi r 0 a. I'm going to kind of put the kappa in front. And then I have 1 over r d dr of r dc tilde dr. And so this is very neat, because the r cancels, and so, sorry, dr. And so I can straight perform the integration without actually knowing C tilde to be, cancel these, to be basically r dc tilde dr at 0 and a. And now if I so this limit is easy, right? If r is equal 0, this term is 0 because r is equal 0, right? But, and that's because we specify the bounding conditions, also evaluating this at r equal a means that I have to evaluate the gradient at dc tilde dr at a, and that we already know by definition, because of the boundary condition, is 0. So this whole term actually is straight 0, which simplifies our equation once more. So it looks like we're almost there, right? So we have a perfect equation for the cross-sectional average of C, which only consists of 4 terms. However, there's this term here that we don't know. And so this is basically what's keeping us from happiness to really identify this term. And so the trick now goes that to find an approximation for this term, the only one that I don't know, but it's important because there could be couplings here, is that I'm going to take the difference of equation 1 minus equation 2. And so would be determined. We want to determine this. So, okay, now if we take the difference, we basically have to copy the terms that are not here. So that's dc tilde dt. This one goes away, plus u tilde dc dz, plus u bar dc tilde dz. And here I actually have this term, right? And then this term as well, because they're functionally different. This one goes away, this one stays, and this one stays too. Okay, so we have a new equation, and we're going to use this to determine C tilde. And then once we determine C tilde, we can plug it in here, and we can, because we know U tilde, which is written here, we can then actually solve for this term. So that's the idea. But as we look at this PDE right now, we cannot do it. So we're going to make approximations now to be able to solve this dynamic in the case that we are really interested. So the first approximation is that we're interested in the long time solution of these dynamics, where long time means that I'm interested in the dynamics on a time scale that's much quicker and that's much longer than the time it takes particles to diffuse along the cross section, which basically means I'm interested in the dynamics of the cross-section average. And so that's T bigger than A squared over kappa. That's the time, sorry, it's a weird A. It's the time it takes a particle to diffuse across the cross-section. And so that means that that the dynamics of C tilde actually average out across the cross-section very quickly, and if I'm interested in the long time limit, this time, the time derivative of C in these long limits, that will actually be zero, because I'm looking at something that is stationary. So the first result of this approximation is that the derivative can be approximated to be zero. Okay, but zero. And the other result is, in addition, that if the diffusion is averaging out quickly, that the variations in C in general should be much smaller than the cross-section average. So that's just kind of like a fluctuation around the cross-section average that I'm interested in. And that will help me to simplify the terms, but I need one more assumption. And so the other assumption that I need is that since C tilde is a radial contraction, it's radial correction only, I will bear in mind that because C tilde is a radial correction, that the gradients in R are much bigger than the gradients along the tube, because that's when they're going to have averaged out. And so this says that dc tilde dr is bigger than dc bar dz. And so this now helps us to go through these terms of these equation here to see which are actually important. And so this is what I specified. Oops. Directly, we're going to this is going to be averaged out, so there's no dynamics there anymore in the long-term solution. Then if I compare now these 4 terms here, then basically the variations in C are always smaller compared to the cross-section average. So comparing those 4 advection terms, the only one that's really dominating is this one here. And the other ones are small due to this assumption. And then here, I said the gradients in C tilde are bigger than the spatial ones, so I'm ignoring that. And so I arrive at an equation in using this approximation that is U tilde dC dz minus kappa over R ER equals 0. And now this is actually an equation that we can solve, right? Because it's an equation for C tilde that only includes radial derivatives for C tilde. And I know my U tilde, so everything is known and I can use this to calculate C tilde, which is what we're going to do now. And so I'll stop here. So we will plug in that U tilde is given by this. Here and R here. And then I can integrate once. Sorry, first integration. Sorry, this is the R dz. Otherwise, it won't work. And so I have plus a constant. I'm going to clean up quickly. Yeah, I already screwed up with prefactors. Let me check. Sorry. A lot mistake. Sorry for that. So this is actually 1 minus 2. So it's a 1, there's a 2 here, and then it's 1/2, and that's another R to the 4th, 1/2. Okay. Okay, so that's the first result. Now I'm going to rearrange to integrate the second time. So we start with U kappa, and then we have 1/2 R to the 3 minus 1/2 R to the 5th A squared minus dC tilde PR, and we perform the second integration, and we arrive at oh, sorry, there's a what about? Oh, sorry. It's like when you do it on the blackboard. So it's dividing by R. I should have done this correctly. It's R up to 3. Yeah, there we go. And then and an integration constant again. So now we have a result for C tilde, but we still have two integration constants. So the first integration constant, C1, goes away because we require the concentration to be finite at R equals 0. And R equals 0 would be infinite at this point. So this goes away. This has to be 0 because C tilde has to be finite at R equals 0. But we still have a second integration constant here. And so the way to deal with the second integration constant is now to take into consideration that basically what I used before, that the cross-section average of C tilde has to be 0. That's just by setup, by design. And so we can just write this down now, which is that because it's 0, I'm going to just drop the prefactors. We integrate from 0 to a r times, and now we're just plugging it out, u kappa 1/4 r squared minus c. And then this gives rise to and it basically only terms that A contribute. So I have here, sorry, let me just 6 and then I can use this to solve, and this has to be z equals 0 by definition, and I can now use this to solve for C tilde. And so my second integration constant then turns out to be, if I rearrange terms, to be a squared u bar over 12 kappa dc bar dz. And with that, I also have the full solution for C bar, which is namely 1 over kappa. And here I'm just omitting really going through all the algebra, which right now is just plain algebra. But I want to give you the full result. And so now we're good, right? Now we have the C tilde, and we know the U tilde, which is basically this term. And so now we can actually perform this integral. I'm not going to go through the details, but I'm just going to give the result, which is if we now plug in this, we can really solve these dynamics. And the important thing to spot is that here I'm taking the spatial derivative along z of this expression, where only C bar is now depending on z anymore. And the rest are regular terms. So basically what this gives rise to is a second derivative in C bar, which is the crucial point if you're going to plug things back together. And so the result of this now is u bar squared a squared over 48 kappa d squared dz bar dz squared. And so now I can plug this in here and I'll actually arrive at what's called Taylor dispersion. And now here you have this term popping up of u squared, a squared, 48 kappa. And this is really the term where the enhanced diffusivity comes into play. And so basically you have to think about it just to understand it holistically. So you have this tube and you have this Poisson profile. And so the cross-section average flow velocity is basically setting your maximum here, right? So the larger your U bar, the larger the gradient of flow velocities along the cross section. And so basically what happens then is that while you have particles like flowing along these streamlines, they do diffuse due to molecular diffusivity. And so they may hop on a neighboring streamline down here, which is going to be faster, or if they hop in the other direction, slower, and thereby the particles spread out more because some are left behind, or some are going to be ejected compared to the reference frame of the initial streamline. And so the steeper the gradient, the more the particles can hop to slower, the quicker they can hop to slower or faster streamlines, and therefore spread out. And that's why it's increasing with the flow velocity, which is proportional to the gradient. And that's the time scale to diffuse across the cross section again. And so what you learned in this exercise was the crucial thing that you should take away from looking at Taylor dispersion is that the key step of writing the dynamics as a cross-section average and the variation around it, like writing what the property that you're interested in, plus the variation around it, and then really checking the scales that are important to you and simplifying the governing equation for this limiting case. Because now that you have an analytic result, you know what contributes to the flow dynamics, even though you're aware that it won't work anymore if you go to, like, say, short, big tubes. But you can always still tackle with better simulations. But this makes your life very easy because now you can, for example, straight ahead solve for the steady state along the tube, and you can also implementing it numerically to look at the transport of particles in the network. It's much easier because when you saw these equations, these movies of a particle spreading out in a network, basically was underlying the dynamics of each individual tube was just Taylor dispersion and not the full equations, which would have taken ages. Another what this is a particularly nice exercise that you will find in fluid physics lectures. Actually, the whole gist of this, we sort of as a you can find probably in textbooks, but in case we also wrote it down nicely in the appendix of a recent paper in PR Fluids. I don't know how you read them. 2019, and there you'll also find out that this approach only works well for the simple Poisson profile. So if you have long slender tubes, but you actually have like space-time contracting tubes, or you have a slip boundary, or whatever, like you have more complicated dynamics, you have a tube that's narrowing. This approach, like the simplistic approach, will fail, but there are other perturbative methods to solve these dynamics, and you can still, like, even if you have more complicated flow fields, you can really solve for the transport dynamics in these long slender tubes, because the simplest assumption, looking at the long time solutions, that's still valid. You just need a little more complicated math. But we also, if this becomes of relevance to you, we really aim to write this down in a pedagogical way that everybody can sort of learn the math and do it on whatever desired example they have. to work this out. And yeah, so and physically speaking, this is really what is important for these flow networks. They're designed to transport particles, and sort of the gist that I meant to give you as well when I was referring to these contractions being coordinated on these large scales. So how does it work, right? So you have this active cytoskeleton that is contracting, and it's somehow keeping a check with the contractions 2 centimeters away to keep a phase relationship. So how does it work? Well, we haven't shown experimentally yet, but based on our understanding on Taylor dispersion, we embarked on the following theoretical endeavor, which was basically to describe the active cytoskeleton by what's well known about actin cortices. Like basically every cell has every animal cell at least, has this cortex underneath its membrane, and so we pretty much know these dynamics. And so what we know particularly about actomyosin is that its contraction is often triggered by a small chemical, namely calcium. And so if you have a lot of calcium, the actomyosin cortex will contract. Furthermore, it's known that if you stretch a membrane, you might have calcium-specific inflow channels that may be activated upon stretching the membrane. So now imagine you have a membrane, a tube, and it's stretched. So calcium flows in. Calcium triggers the contraction of the tube, but then calcium is also quickly recycled into vesicles again, and so the tube relaxes until it's stretched enough that it contracts again. So this is basically well known in the literature. And so you really can well describe the dynamics of a tube as a function of this contractile force, where now the contractile force is coupled to calcium flowing in due to stretch or being cycled within the bulk of the cytoplasm. And now the only key idea that we put into this is saying that calcium is also being advected with the fluid flow. Calcium is being transported with Taylor dispersion. This idea alone is sufficient to explain these large-scale coordination of contractions, because calcium is being transported with the fluid flows, and now it couples onto the contractions and generates the flows that it's driving on. And thereby, you can really generate this large-scale scaling, which is where the key point is really that you have these long-ranging flows that are feeding back into the scaling itself. And so that's another way to underline that it's not just maybe wonderful for, like, you know, getting nutrients spread in throughout a network to have Taylor dispersion, but it's really a mean of communication, that signaling molecules are being transported with the fluid flow and they're coupling back to the tube state and thereby coordinate the dynamics of the whole system. And so that's why this equation has very deep meaning, and it's wonderful to keep in context when in general looking at flow networks. That would be my gist for today. Thank you. Happy to take questions.
Thanks, Karen. Questions? Yeah.
I didn't get why the coefficient related to the log R term was kept 0.
Oh, this one.
Yeah.
There. Yeah. So so this is the equation. So if I just move the second constant over here, that's the equation for C tilde. So if I now evaluate C tilde at R equals 0, this term will be 0. This is a constant, but here I have a diverging term, right? It becomes minus infinite.
Right.
And I mean, by concept, C tilde should just be a variation in radius and it should not diverge. And so that's why this term has to be, this constant has to be 0.
Thanks.
Sure.
The questions. Okay, Karen, could you give us an idea of what you're going to do tomorrow?
Tomorrow we will actually embark on flow networks, and we're going to solve flows in networks. So tomorrow will only be Blackboard, unless you request otherwise. We're going to talk about principles of how flow networks are organized. We're going to talk about Murray's law and network structures that form under minimal dissipation, and also other tasks of flow networks, namely transport and absorption, which is then going to be a little bit of a reminder of what we saw on the Blackboard today.
Great.
But we're going to go basically from a tube to networks.
Okay. No other questions? All right, so let's thank Karen for a very nice talk and